Good morning, I'm Simona Daniele from the Department of Pharmacy at the University of Pisa. Today I will present you our study on sex-specific blood changes in neurodegenerative proteins and antioxidant responses to regular physical exercise. I will walk you through the rationale, methods, key results, mechanistic insights, and implications for future research. Let's start from the background. As you probably know, dementia and other neurodegenerative diseases are a growing global burden, and understanding modifiable protective factors is urgent. Among these, Alzheimer's disease is the most common form of dementia, in which extracellular amyloid beta plaques accumulate in Alzheimer's disease brain and disrupt neuronal function, thus promoting cell dysfunction and apoptosis. Alzheimer's disease arises from multiple interacting factors that compromise neuronal health. Genetic risks such as APP, presenilin, and ApoE sequences can alter amyloid processing and lipid metabolism, while blood-brain barrier dysfunction, mitochondrial failure, and obviously oxidative stress further impair the clearance of toxic protein and promote neural inflammation. Environmental and lifestyle contributors, including aging, cardiometabolic disease, and traumatic brain injury, modulate this pathway and shape individual risk. Importantly, sex differences influence vulnerability. Indeed, females show higher dementia risk and more severe AD pathology at older ages, whereas men have greater predisposition to Parkinson's disease. Moreover, not only amyloid beta is involved in Alzheimer's disease pathology, but also hyperphosphorylated protein tau can detach from microtubules, destabilize axonal transport and forming neurofibrillary tangles, and also alpha-synuclein can aggregate into misfolded oligomers and fibrils, further disrupting membrane integrity, vesicle trafficking, and proteostasis. What about physical activity? It is a promising intervention, but the biological responses to exercise appear to be different by sex. Moreover, there is a big heterogeneity of evidence, and meta-analyses show a variable cognitive outcome from exercise due to different samples, protocol, and participant gender. Our study asked whether long-term moderate exercise can produce sex-specific changes in peripheral markers linked to neurodegeneration and antioxidant defenses. Our study therefore compares peripheral accumulation of neurodegeneration-related proteins and antioxidant epigenetic response to long moderate exercise in premenopausal women versus age-matched men. The goal is to identify sex-specific molecular signatures that could explain why exercise can reduce certain neurodegenerative markers differently in women and men, and to inform tailored prevention strategies. What about our study design and participants? We enrolled more than 100 healthy adults, 60 females and 60 males. Female participants were premenopausal and do not on hormonal therapy. Athletes were defined as performing at least 150 minutes per week of moderate aerobic exercise for 10 or more years. Samples were collected at least 40 hours after the last exercise bout. As you can see from this table, age and BMI were comparable across groups, and as expected, athletes reported high perceived exertion, that is the Borg 15 scale, and had higher circulating testosterone levels for men subjects, while female had higher estradiol and lower testosterone than males, consistent with the physiological sex differences. What did we measure? We measured erythrocytes level of A-beta, total and oligomeric alpha-synuclein, tau by specific immunoenzymatic assay. As I showed you before, in plasma we quantified estradiol and testosterone, but also total antioxidant capability using the TOSC assay against 3 different radicals, and finally different circulating epigenetic markers, MIRA and DNA methyltransferase, of which I'll tell you later in the next slides. Erythrocytes were chosen as peripheral proxies for protein accumulation and redox status. What about plasma antioxidant capability? Overall, athletes showed higher plasma antioxidant capability than sedentary subjects, as shown in previous paper. However, sex differences emerged. Indeed, in men, exercise increased scavenging of all the radicals. In contrast, in women, exercise selectively enhanced scavenging toward peroxy radicals only, and there were no baseline sex difference in antioxidant capability, thus suggesting a sex-specific modulation of antioxidant capability by moderate. And what about erythrocyte A-beta and tau levels? We showed that at baseline, females presented higher erythrocyte A-beta levels than the males. And in contrast, males presented significantly higher levels for baseline erythrocyte tau than females. Exercise significantly lowered A-beta in women and with unchanged tau, and significantly lowered tau levels in men with unchanged beta. So, suggesting a sex-specific neuroprotective adaptation to regular physical activity. At baseline, females presented higher erythrocyte total alpha-synuclein levels than male, but these levels were not modulated by physical exercise. Moreover, oligomeric form of alpha-synuclein were similar across all the groups and were not modulated by physical exercise. After this, we move to examine epigenetic circulating markers. Among these, NRF2, that is a master regulator of antioxidant defenses, which activates genes that detoxify reactive oxygen species and support proteostasis. Its activity is modulated by chromatin regulators and can be indirectly influenced by microRNA and histone modifiers, thus altering transcriptional programs for cytoprotective genes. Moreover, we measure histone deacetylase 6, that is a deacetylase that regulates protein quality control, autophagy, and clearance of misfolded proteins. Moreover, we measured the plasma concentration of circulating specific microRNA, such as 153, that is a post-transcriptional regulator that is linked to NRF2 axis and is reported to increase with oxidative stress. It acts by repression of mRNA targets that control antioxidant and autophagy pathways. Moreover, we looked at MIR-195 that regulates the amylogenic processing and has been reported to have neuroprotective and anti-amyloidogenic effects by modulating the translation of key neurodegeneration-related genes. We obtained that exercise significantly enhanced NRF2 levels in athletes of both sex versus sedentary subjects, and exercise also significantly lowers the levels of histone deacetylase 6 in athletes of both sexes versus sedentary subjects. Moreover, we obtain a significant increase in athletes of MIR-185 and a significantly decrease of MIR-153. What we can say? We can say that the combination of an enhancement of NRF2 and a decrease of histone deacetylase 6 indicates a cellular environment favoring detoxification, autophagy, and cytoskeletal stability. Moreover, the enhancement of MIR-185 suggests a regulation of amyloid precursor protein toward a non-amyloidogenic pathway, and finally, the decrease of MIR-153 indicates the release inhibition of NRF2, amplifying antioxidant defenses. Now I'm going to show you some selected correlations that we obtain among parameters. As you can see, in male we obtain a positive significant correlation between the perceived level of physical activity, that is the Borg scale, and the plasma antioxidant capability towards all the measured radicals. In contrast, in female, we obtain a significant direct correlation between the Borg scale and the plasma antioxidant capability versus peroxynitrite radicals only. That is that a higher perceived effort, the RPA or Borg scales, correlates with stronger antioxidant capacity, but a broad response was obtained toward a selective peroxynitrite scavenging only in women. Moreover, in men, higher testosterone levels correlate with the lower tau, aligning with the male-specific reduction of tau that we observed in athletes. Consistent with this data, higher estradiol associate with lower erythrocyte A-beta levels in women, supporting a female-specific hormonal protection against beta amyloid accumulation. Finally, looking at epigenetic markers, we noted a significant positive correlation between testosterone level and NRF2 in both sexes, indicating that testosterone can enhance NRF2-mediated antioxidant and proteostatic responses. Finally, in men, we noted a positive correlation between testosterone and miRNA 185 levels in men. We know that this is a microRNA with anti-amyloidogenic and neuroprotective roles that can be supported by the levels of testosterone in men. So to conclude, we should say that our cohort focused on premenopausal women, thus limiting generalizability to older or postmenopausal populations, and that erythrocyte measures are peripheral proxies that may not fully reflect central nervous system pathology. Nevertheless, to summarize our results, we showed that females had higher erythrocyte A-beta and synuclein at baseline, and exercise reduced A-beta in females and reduces tau in males. Moreover, exercise enhanced antioxidant and autophagic signals through the enhancement of NRF2 and miR195 and by reducing the levels of histone deacetylase 6 and miRNA 153. And as antioxidant responses to exercise were broader in men, and a peroxynitrite selective effect was evidenced in women. Finally, we showed that sex hormone, testosterone, and estradiol correlated with these molecular changes. Finally, I would like to thank all co-authors and collaborators listed in the manuscript, the institutional partners, including the Department of Pharmacy and obviously the Sports Medicine Unit at the University of Pisa, all the technical staff, the laboratory personnel, and most importantly, the volunteer participants in the athletes and sedentary subgroups. And obviously, I thank you for your attention and I welcome your questions.